Welcome to week 6 of your online organic laboratory. This week we're going to be discussing chromatography, which is a technique that can be used in several different ways. Mainly today it's automated via instrumentation, either gas chromatography, liquid chromatography. You might have heard of instruments called GCs or HPLCs or just LC. That just stands for gas chromatography or liquid chromatography. And basically it just comes down to this is a specific technique used to separate different constituents of a solution or a mixture that you might have. Expanding a little bit more about chromatography, as I said in the intro, this is really just a method to separate compounds. The big key point here is it's all equilibrium based. And we use the separation techniques to determine a difference in equilibrium between having some solid phase that's immobile. Either it's going to be a plate, a silicon-based plate or aluminum-based plate, or it can be a column inside of an instrumentation, like a GC or an LC. A column is just basically a high surface area tube that you run your sample through, and based on interactions and equilibrium between the solution or mixture that you have and the walls of the column, some of the compounds that can be found there elute or move through, whatever you want to call it, slower based on either polarity interactions or hydrophobicity reactions, stuff like that. But the key here is chromatography is what we're using now, and any instrumentation is either going to be a moving liquid or gas. And that moving liquid or gas is generally going to be propelled either by some type of electromotive force in terms of electron flow or solvent flow, that type of thing, or it's going to be propelled through a column in the terms of instrumentation, and it's generally propelled through by either a null or a shear gas that's very neutral and will be reacting with anything. So it's a known quantity, so you can subtract it out of your baseline. And generally you use something like a nitrogen or argon that are generally non-reactive and fairly cheap. That's the important part. Here we have a simple little chart diagram about the separation of two components. We'll label them A and B. And you have, they could be liquid or gas, doesn't matter. But while they're in solution or mixture, A and B are at equilibrium between each other. They're happy, they're moving around equally, everything's nice and good. However, when you put them into this column or LC plate, like we're going to be using today, there's interaction between both A and B with your solid phase. In terms of the diagram at the bottom, the shaded blue area is your liquid or gas phase that's going to be moving across this solid phase, which is the white on the bottom. So as A and B interact and bump into the interface between your moving phase and your solid phase, B in this circumstance is actually stronger in terms of its attraction to the solid phase than A is. So basically what that does is every time B bumps into that solid interface down at the bottom there, it sticks there for a second, or maybe longer. It doesn't really matter, just a small instance of time. Whereas A generally isn't attracted and either never hits the sides or bounces off real quick and never actually sticks there. What this causes is A actually comes out of solution or elutes out of your liquid or gas phase quicker than B. Now, with what we're doing, it's called a TLC or a thin layer chromatography, use on an alumina or a silicon plate. And the specific compounds in your solution will slowly elute up the plate as you're going to use a solvent, and that solvent is slowly going to climb up this plate. And you put little dots, which we'll talk about in a second, of your compound, and you watch how they elute or move up. So in this circumstance, you would have two dots of A and B. Your solvent line would move up the TLC plate, and because B would be more attracted to your solvent, it would go slower. It wouldn't go as fast, while as A is constantly pushed up. So it's the same basic principle between using a TLC plate or using an instrument. And for the instrument, The area in this diagram that's shaded blue would basically be a column. It's a very thin tube with lots of surface area and a specific coating inside. You pass your solution through it. A is not attracted to the lining. B is. So as your argon gas or nitrogen gas pushes it through, B is much slower because it's interacting with the sides and the inside of the column. Therefore, A comes out earlier, is detected first, has its unique peak. B comes out later, has its own unique peak, and you can separate and tell exactly what you have in there. And if you have a solid phase or a column that's long enough or can attract a specific compound well enough, you get a solution like this. It's the same thing. You got separation, two components A and B, and here with enough time or enough distance traveled, you can get a complete and total separation where B is being attracted so much and A is not being attracted at all that A is completely eluted out of solution and is removed totally. And there's some processes that can do this. If you kind of need to know what you have, it's not very good for using an unknown if you want to do this type of situation. But there are some instruments that can actually separate compounds based on GC or LC, and most of those use something called thermal conductivity, which basically passes that specific elute or component of your solution over what's called I believe it's a Wheatstone bridge, which is basically a triangle of resistors it's very sensitive to changes in temperature. So as those compounds pass over that Wheatstone bridge, you get a slight difference in thermal conductivity based on the molecule. And that thermal conductivity is a physical property that's unique to each molecule, so you're able to use database lookups and stuff like that, know exactly what you have. But using that, you can separate basically something in a solution if you're looking to keep one part of it. So this is an example of something along those lines. Now moving on to thin layer chromatography, which is what you would be using in this lab, because most of the instrumentation is, honestly, it's expensive and they don't want undergraduates messing around with them too much. But thin layer chromatography, of course, came about before all these instruments and is still used if you just want to do a quick experiment to check if something's still in solution or if you have any, extra components in there you're not expecting. This is still used constantly, especially in organic or synthesis labs. Most of the grad students here can tell you they've got boxes and boxes of these TLC plates, which, like I said, is thin layer chromatography. But talking about those a little bit, your TLC plates, the solid phase is the plate itself, and like I said earlier, it's either some type of silica or alumina. And the silica gel is usually silicon dioxide that's hydroxylated, or your alumina is aluminum oxide, Al2O3. And basically, these are pressed onto almost a foil-like substance, uh, and they're very fragile because it's a powder is really what it is, but it's pressed at high pressure, and just like a pill is basically a powder, but they press it at super high pressure so it stays at a hard pill form. It's the same type of thing. These aren't pressed that hard because you'll destroy the base of it, but they're very fragile. So anytime you use them, they're going to be supported on either glass, plastic, or most of the time it's a thicker grade aluminum foil is what most of them are. But because they're fragile, when you handle these in a real lab, you got to be very careful with them because even the slightest bump, you can crack it and that ruins the thing. And they're not really all that expensive. It's just, you could lose data, so you got to be careful with them. And the number one key when using TLC, and I'm sure there's going to be a question about this, is you never put your finger on the surface of it. Because even though your finger or your glove, which you should always be using gloves in the lab, of course, might look completely clean and completely fine, you've got oils, you've got contaminants, you've got dust and dirt, and that can mess up and show up as something in your TLC when you run it that you didn't expect. So always look out for that. Never touch and it's going to be white. It's going to be white, like pressed powder on this thing. So never put your fingers on that white substance. Always hold it from generally the sides. kind of clamp it like use your thumb and your forefinger as like a claw and clamp it on the sides. Hold it that way. That's the best way to hold it. So solid phase is either the silica or the alumina. And the movement phase, because we don't have any shear gas or any instrumentation, what we do with TLC is you use an organic solvent or a mixture of solvents, depending on if you want to, see how things work a little bit better, more intricate of a system. So in terms of TLC, basically your solvents are going to be tailored to what you think you have. For instance, polar solvents generally cause rapid movements because as that polar solvent is moving up the TLC layer, it's more prone to pull things with it because hopefully you all know if you have a polar substance, it's got specific areas of electron density and electron sorry about that, my mic cut out. It's got polar substances have specific areas of electron density and other areas where electrons have been withdrawn from. So you got partial positives, partial negatives. So as the polar solvent is moving through the TLC solid phase, it's actually pulling anything else that is polar or ionic or through and carrying it with it. So it's actually like an electromotive force. It's a driving force. It's you're pulling it with electrons. It's a chain that goes on the whole system. So, and like it says in the bottom, there's a strong attraction of polar solid phase itself because if you notice, the solid phase is either silicon dioxide or aluminum oxide. So we've got some type of either the aluminum oxide is basically an ionic compound, and the silicon dioxide has those oxygens are very highly electron withdrawing. So we've got polarity there, and also silicon has its own lone pair when you do the Lewis structure of silicon dioxide. So basically, anything that's if you're going to use a polar solvent for your movement phase, anything that is less polar or non-polar is going to be excluded from or uh, not eluted up the line. It's going to stay down where it initially started, and it's kind of hard to see. But once I start showing you actually the way it works, you'll kind of understand what's going on. Hopefully, a little bit better than now. Here we have a great example of what a normal everyday lab use TLC plate looks like. Um, the plates themselves are generally a lot wider, but you cut them with high quality scissors. You say you don't need to use a huge plate for one drop. But right here, this is an example of a fully eluted TLC plate. And for this one, they actually just put one drop of black ink, the standard black ink you find in any pen on there. And the first thing you should notice, of course, is we have a whole different hue of colors. That's all the constituents of black ink. Black ink isn't just black. It's a mixture of multiple colors that when the eye sees it, it just perceives it as absorbing all light frequencies, so it shows up as black instead of reflecting. So that's what it is. It's also a very good way to show TLC and how the system works because it does separate out like this. So first thing to note is the TLC plate itself. There's the white, very thin, powdery layer covering it. And if you look on the left side, there's even an arrow pointing to it. You can see that underneath is that heavy aluminum foil as its base. The markings on it, it's actually because of the powder. If you are very fine with a pencil, don't use a pen because the ink will come out. But if you use a pencil, graphite won't travel through the solvent. So lines are drawn with pencil, and you can mark it with pencil and show where certain things are. So always use a pencil if you're going to mark your TLC plates because you don't want inks interacting with it. So that's the typical TLC plate that's been eluted. And the chamber they use and what you would actually use in the lab too is you just get a normal beaker, and in the bottom of this beaker will be a couple inches or so of your solvent, and then you're going to cover it with a wash glass. You cover it with a wash glass because solvents are generally have a higher volatility, so they'll start to evaporate. And you want to keep your solvent in solution you want to keep your solvent in a liquid form as much as possible instead of a gas. So that's why you keep the wash glass over your beaker so things won't evaporate. But here you can notice on the bottom left of that TLC plate, there's that one big, kind of multicolored greenish, bluish dot down there. That's actually, and then there's a line through it. That line is your starting line basically, and you would mark that a couple inches above the base of the plate, and then you would put one drop of black ink right in the very center of that, and you're going to dip that into the solvent, the bottom side first, making sure that the solvent doesn't actually come up to that line because you don't want it to wash down into the solvent. You want it to lute up. So it's going to be about halfway up, probably, your solvent line from the base of the TLC plate to that first line. It's going to be halfway up. And over time, your solvent is just due to physical interactions, electromotive forces, because the plate itself is polar, the solvent is polar. Solvent is slowly going to be pulled up this plate and make it wet, and it's going to go all the way up to the top. Now, as that solvent line, that's what we call a solvent line, because you can actually see it gets a little darker, a little less kind of grayish instead of in terms of white when you're doing this. As that solvent line goes up, it's going to hit that ink spot, and anything that can be dissolved or interacted with that polar solvent is going to be pulled with the solvent as it moves up the plate. And that's why you see the smearing and the different things. So you got all those different colors that are being pulled up, and as you can see, the very, very light blue starts first, and then you get into greens, and then you get into the indigo colors, and then you get some weird pinkish and purples and reds and yellows. So the higher up on the plate that specific marking ends, that means it's more attracted to your polar solvent because it's easier to move around. So basically you can tell just from these dyes that are using here, that yellow, orange, and red dyes are more polar in nature than your blues and greens, because the blues and greens are closer to the starting point, whereas the reds, orange, and yellows are higher up. And that's basically the simple version of TLC. Here we have another example of a fully eluted TLC plate after the experiment's already been done. On this one we have a mixture of ortho, meta, and para compounds. These are nitroanilines. Aniline is basically just a benzene ring with a NH2 group substituted primarily. And then the nitroaniline just means you have a nitro group, a NO2 group attached. Ortho is directly next to an NH2 group. Meta is 2 away, and para is opposite. So all these will have different interactions, even though it's the same structure basically, just different little isomeric conformations of this thing. So if you see, notice here you've got ortho, meta, and para are the first 3 dots, and you can tell that the ortho is a lot more polar than your other 2 because it's drawn up the TLC plate higher, and that's basically what you should expect because your NH2 group is slightly, it's got polarity because you got lone pairs on the nitrogen, and then you got 2 hydrogens sticking off of it, so those are electron withdrawing. For the lone pair, there's a higher density right there, and the hydrogens themselves are generally partially positive for the most part when you see them. And then the NO2 is highly electron withdrawing because of the oxygens and a lone pair on the nitrogen. So when you have both of those together, you basically have the area around the NH2 group and the NO2 group. That whole area is kind of electron withdrawing. It withdraws it. Whereas looking at the meta and the para, you're starting to separate those out. So you're getting different areas of electron density, and they basically cancel each other based on symmetry, which you can see from the para version that it is a little polar because the elute goes up a little bit on the plate, so it's going to be pulled up. But because your NO2 and your NH2 groups are adjacent to each other and symmetrical, they kind of cancel each other out. The NO2 is actually more polar than the NH2, so you still have some polarity, but there's canceling there. That's why it doesn't move as much as the ortho, for example. And another thing to note here is the very last dot or group of dots was an unknown mixture of some of these compounds. And if you notice, you can basically line it up. There's 2 big areas of yellow smudges, and it's basically lined up directly with a para and an ortho. So you can do, if you know what certain things are and you take a mixture, you can compare it and say, all right, this was basically a mixture of ortho and para. nitroaniline. That's another way to use TLC. Another example of TLC usage is the identification of colorless compounds. Um, when we're talking about organic compounds specifically, a lot of organic compounds can be activated via UV light. It excites the electrons, causes a fluorescence. So basically what you can do in this case is when you run a TLC, and if you don't actually see dots or smears, if you don't have a physical visible light excitation of coloring, you can illuminate it with a UV light and you get that green glow. The green itself glow is from the alumina, the silica gel, but that lesser color, the black or absence of as much glow, that's where your spots are going to be. And so that's what you can use if you don't actually see your spots. And of course, like I told you earlier, there's a definite pointer here that's holding it incorrectly. Your fingerprints are very bad. You only hold the plate by the edges. So expect a question like that on your exam. Moving on to actually doing a TLC experiment. These are the basic steps that you would use for every time you do it. It never changes, so it's pretty simple once you got it once. So the very first thing you would do is your cutout TLC plate that your TAs or your lab director would give you. You draw that little line a couple inches from the bottom, and then you spot it. Depending on all your compounds of interest or unknowns, you can have multiple spots, but you need to make sure there's a good separation between the two because when the solvent hits them, they kind of tend to spread a little bit. Not a ton, but they do spread, so you want to have enough room for everything to move, basically. Once you have the plate spotted, then you're going to place it into a beaker holding your solvent. And the important note is the solvent can't touch any of the spots. It has to be well below. Generally, you don't want it any more than halfway to the dots. So if you have too much solvent in there, take some out before you put the plate all the way in there. Once the plate's all the way in there, you're going to cover it with a watch glass, and you're just going to watch the solution, the solvent, move up the plate, and allow it to move all the way up to the plate, but never to the top. Leave about an inch from the top. You don't want it to go all the way to the top, because then the solvent can start going back down the back of the plate and start pulling the dots down. So you don't want that. So, and it's easy to see when you're looking at it because the alumina or the silica composite on top, as the solvent moves up, it gets darker and you can actually see it slowly moving up the plate. And depending on what type of solvent it is, if it's really polar, it moves pretty quick. So sometimes you have to take it out pretty fast. But right before it gets to the top, take the watch glass off, take the plate out. Remember, only touching it from the sides. And once it's dried, you basically leave it in a fume hood on a watch glass and you let it dry out. And then once it's dry out, you're able to circle the dots of interest and take readings, use UV to see if there's anything you're not able to see under visible light. And remember always to use a pencil to mark any of your dots. Now, in terms of identification of your dots and trying to figure out what things are, there's a technique that uses something called a retention factor, which is basically just how well the solvent or the plate can attract those molecules of question and allow them to be either retained or not in the TLC plate. So basically, that's why you're using a pencil and marking everything. The first thing you're going to mark is the top of how the farthest up the solvent line is. You basically would just draw a line across once you took your plate out to mark the highest that the solvent went up the plate. Then you would mark each specific dot and label them with a number, and then you would label your starting line as number 1. So basically what you're doing is you're just forming a ratio of how things went based on how far the actual solvent went. And that's basically called a retention factor. So you would measure the distance from your starting line to the top of the solvent elution line. So basically the distance between number 1 and number 4. And that's D14. Just call it that, simple. Then you're going to measure the distance from line 1 to your dots, how many ever you have. And this, they're only using 2 for simplicity, but the distance from line 1 to 2 would just be D12. The distance from line 1 to 3 would just be 1, 3, D13. So your retention factors are for instance, for spot 2, the blue spot, you take the distance from 1 to 2 and you divide that by your distance from 1 to 4, the elution line. And that is your attention factor for that specific spot marker. And D3 is the same way. It's the distance between 1 and 3 divided by the distance between 1 and 4 for that retention factor. So each specific dot has its own specific retention factor. And you can do TLCs with known compounds that you expect might be in there, stuff like that, if you have a hunch. And you can compare retention factors. And in this case, it's always going to have the same retention factor no matter the size of the plate you use, as long as you use the same solvent. Now, in terms of a tool to spot your TLC plates, generally what used to be done is you make a spotting capillary. You would just take a capillary tube, which in general labs you find them all over the place, and you'd use a heat source at the very center of the capillary tube, and you slowly start pulling the tube apart as the glass starts to get weakened. And as you slowly pull it apart, it gets thinner and thinner, and until it finally breaks right in the center, and you get a very thin, tapered, sealed glass spotter, basically, where you can have a very small dot of your compound of interest. Generally, when you do this lab in person, we show you how to do it, and we let each person do one, but we don't really mess around with this too much, because nowadays, you can buy pre-made capillary spotters and stuff, basically the exact same cost as the capillary tube. So why deal with flame? Why deal with burning yourself in molten glass? So, but if you ever see it, that's how you used to make a spotting tube. Just another few small points on your choice of eluting solvents. Like we said earlier, the more polar your solvent, you're going to get faster elution and generally a greater separation of your dots, separation of your compounds. So a lot of times you're actually going to use a mixture of solvents, a mixture of different polarities and stuff like that, just to make sure. Because if you have a solution that has 4 or 5 things in it, generally it's going to be where 1 or 2 of those might not even interact with your main solvent at all. So you would have to put other solvents in there that will interact, but don't interfere with your other solvents. So you generally make a mixture, and this just in basically everything in organic chemistry comes down to the type of solvent you use. And there's a couple tricks just from knowledge and experience in this, but most of the time it's trial and error. It's just to see, unless you know specifically. So if you have any questions on what solvents you would use, it's generally going to be something along the lines of you have 3 compounds in a solution, and these 2 are highly polar and are interacted to this, but your 3rd compound isn't, but it's capable of interacting with this other solvent. So you would make a solvent mixture of 2 different solvents to elute everything out of your spotting dots, so you would be able to see each individual component. Here we have just a simple chart showing this expected elution value for most organic classes of solvents and organic classes of actual constituents of your unknown solution, or known, whatever you're looking to figure out. So looking kind of at the bottom first, that's where the solvents are. So like we said, increasing the polarity, they're more powerful. So if you notice, starting at the top of the bottom section, alkanes at the top, they're not polar at all. It's just hydrocarbons. So there's no polarity. Then he goes to something like toluene, which is a benzene ring with a methyl group substituent. So that's basically, for most intents and purposes, non-polar, but it's more polar than an alkane itself. And then you go into things like halogenated hydrocarbons, where it's mainly hydrocarbon, but you have a halogenated substituent that's an electron withdrawing group. And then you go into your compounds like ethers and ethyl acetates and acetones. And at the very bottom, you get things like alcohols and acetic acids, or any type of carboxylic acids, which are going to be your highest, most polar, generally, of your basically standard solvents for organic elution. And then looking at the top, it's basically the same thing. You're out if you have a substance that has alkanes as a liquid in it, those are going to be very nonpolar, so they're going to have uh, basically no interaction. They're not going to move with your polar with a highly polar uh, solvent. So if you know you have alkanes in there and you want to separate them out just to see, different types of alkanes, you're going to have to use a much less polar. Generally, you would use something else, some type of alkane like a hexane or petroleum product to solvate that out and elute it. Otherwise, it's not going to move in a polar because it's not going to interact. Same thing, it just goes down alkanes, alkenes, then you get ethers, halogenated aromatics, and then of course at the bottom you're going to get alcohols and carboxylic acids. So it's the same thing. It's just about choosing the right solvent for doing the job and separating what you might have. And like I said, if you have an unknown, you're just gonna have to try a whole bunch of different solvents. And it might be an example that I actually had in lab is I was making some dyes for catalytic conversion of water, and the dye I made had a bunch of we'll say ionic compounds in it and some organics as well. So I had to do a bunch of TLC to see of all the side reactions that were being made when creating the dye. And I noticed I had a bunch that went with polar, but I had a few that weren't. So I ended up making a mixture of 4 different solvents to be able to pull everything out at once so I could track and make sure everything was out of there. So that's just a type of thing you might be doing with this type of situation. Now, for your actual lab this week, we had a lot of introduction here, but it's a new topic and it's kind of, you know, broad, so we had to do a lot of discussion. But for the actual lab, it's a separation of analgesic tablets. And analgesics, if you haven't heard that word, it's just a painkiller. That's all they are. So your standard painkillers, you've got aspirin, you've got acetaminophen, which is Tylenol, um, phenylephrine, ibuprofen, naproxen, caffeine's actually analgesic as well. Uh, so naproxen, like naproxen sodium or salicylamide, is some of the earlier things. But for your specific lab, you will be given 3 to 4 different versions of these. They're already going to be dissolved, crushed up and dissolved, and made into a solution where they're fully dissolved. And so we just have a picture, some diagrams of all these. And as you can notice, that basically every one of these compounds is polar in some form or fashion. You've got extremely polar ones, like for instance ibuprofen, where the electron density is mainly all congregated at the very end of the molecule. Aspirin is fairly polar. You got phenylephrine. We've got hydroxyl group and an amino group. So those are very well known electron withdrawing groups. Acetaminophen is basically the same thing. Caffeine is slightly less polar. You got a bunch of lone pairs of the nitrogen and withdrawing groups from the oxygen, but because of the way things are situated, they kind of bump into each other a little bit, so it's not as polar. But basically, all of these compounds can be eluted out with polar. So for this type of thing, you can separate all of these only using only one solvent. So that's what we're going to do. And because they're all slightly different, they're not all going to elute at the same rate as all the ones. So some of the dots are going to be higher than the others, and you'll have a nice cascade. So it's a good experiment just to test out using a simple TLC system. So one of the things you will be doing for this lab is when you would do it in the actual lab, you see what's the fastest moving spot for the analgesics on the TLC plate and the slowest moving. So your fastest moving will of course be the most polar, and the least polar or the slowest moving is basically you can use that data because you only have 3 or maybe 4 at the most. You can relate these to the possible structures you would have. So basically, when you do this, you'll know what you have. You'll know this is this. You'll have this compound, this compound, this compound, and so you'll give it 3, and then we'll give you an unknown or a mixture of it, and you have to determine that based on what you already have on that plate. So you can look, okay, I've got these 3 known things, and you can go back to the previous slide and say, all right, this was the highest, so it's the most polar. So you would look at the compound and determine, all right, which one of these 3 compounds is the most polar, which is the least polar, and then the middle one fits in. And so you would label them as such after you're done with the initial TLC. And then you can do the unknown, and you can compare it and see if those same dots show up, and darker or lighter, or if there's multiple dots in your elution, so you can say you have a mixture of it, or you can give a really, really vague, uh, basically ratio of it. If you look at the dot and it's half as dark as your initial known, you can say you have half as much concentration. You can't say that, legitimately, but you can say as a table, as a napkin math type of thing, you can say, based on what I see, it looks to be about as half as concentrated from the actual original you looked at. So basically, that's a good way of relating structures based on elution, based on how far it elutes up means it's more polar. If it's closer to the starting line, it's less polar. And what you're going to look for in this specific example of analgesics is multiple OH and NH groups are strongly uh, electron withdrawing. Anytime you have a carboxylic substituent that's very strongly withdrawing. But then you also have to take into account, is there symmetry? Are things across from each other? Are they close to each other? Are they farther away? That's going to play in there too. So it's all general polarity rules that you have learned from your organic lectures. All right, and when you would do this in lab, just a reiteration of the typical spotting for your TLC plate. These will be cut out for you ahead of time by your TA. We don't want you messing with it. They get really messy if you do them right. And you're only going to use pencil, of course. So um, from the bottom, you make a very light pencil line to be your starting elution line or spotting line. And then you make very light marks on where you're going to put the spots, just to know, because you might have some compounds that don't show up. So as long as you put those little marks on little dots on the bottom based on how many of your compounds you're going to test, you'll know that something's there. You might not see it, but something's there. And if you have enough room below those dots, you can even write out what is supposed to be there. It helps if you got multiple plates you're doing. And then when that's done, you're going to spot your plate on those little dots, little pencil dots you drew. And the key thing here is to ensure you have the smallest spot as possible. You don't want a big spot, because that's going to give more surface area to the spot. It can dry out before anything happens. You can also get spread to other dots or down or above the line. So you want as small of a dot as possible. So once you have that done, we're going to have the leftmost spot be your unknown if you're given one for your specific reaction. So leftmost dot is your unknown, and then those other three dots can be used for your known compounds. And of course, if you can, either write the chemical formula below it or just an abbreviation to let you know what they are, just so in case you forget. And of course, if you have less than 4 total, just space them out. Just make sure everything's nice and spaced. But this will let you compare your unknown to your knowns, and the general ratio and some basic information you can get from that. Actually doing this in the lab, depending on your section and how much time you have, some of these can take a while to elute, and then you got to dry them and then mark them and check them with UV. So it's most of the time it's not a very quick process. So whereas those of you that have the book, it shows you might test a whole bunch of things. In the actual lab, you're only going to do maybe 2, based on the section you're in. If you're in a major section, you can do 3 or 4, because the lab's basically twice as long. But if you do 3 unknowns, 3 knowns, 3 with unknowns, you would just do a plate for each of the unknowns. And if you were going to do like 4, you can separate it. You can do a plate for each unknown, and only have a couple of knowns on it. But basically, it's just for efficiency's sake. It's not anything. It's not like a guiding rule. But the key thing to take away from this is these aren't, for the most part, these aren't very fast tests, because you got to get everything set up. You got to get the cut plates and label them, and be very careful with the tiny drops. And it's always the first few times you do it, you always mess something up, so you got to start over, or someone always accidentally knocks something onto the plate or drops the plate and it cracks. So this is a longer process, so you just always try to make it as efficient as possible. For our solvent choices, the elution solvent for your analgesic solutions, your TA will have made, if you were in the lab, a mixture of all your solvents, and they'll be ready for you. And everyone would be doing basically 3 plates in their groups. So the first plate, the elution solvent is going to be a mixture of glacial acetic acid and dichloroethane. Now, glacial acetic acid is basically as pure of an acid as you can get, of acetic acid that you can get with this very small amount of water. I believe to be called glacial, it has to be at least 80% acetic acid. So even though acetic acid is the main component of vinegar, it's a very, very weak acid. Glacial acetic acid is very dangerous, and it gives off a lot of fumes. So that's a safety issue with that anytime you work with glacial acetic. So that's for your first one. It's going to be glacial acetic acid and dichloroethane. And if you notice, volume to volume ratio, it's 1 part glacial acetic to 12 parts dichloroethane. So it's mainly dichloroethane. So that's going to be the main constituent for that one. For your second elution solvent, it's going to be a 4-part mixture, and by volume it's 5 to 1 to 1 to 2. And the 5 is going to be the dichloroethane again, and then you're going to have 1 part acetone, 1 part 95% ethanol, and then your final part is going to be hexane. So that right there should tell you that, you know, the dichloroethane is fairly polar, acetone is very polar, ethanol is very polar, hexanes are nonpolar. So that's a good solution if you have got a whole bunch of stuff in there. You'll get a good separation between your compounds. And then finally, your last plate is going to be another 2-part solvent for elution, and this is going to be a 25 to 1 volume by volume compound. And this is going to be ethyl acetate, which is fairly weak polarity, and then glacial acetic acid, which as a carboxylic acid is very strong polarity. So this is a good solvent solution if you have a mixture of strong versus weak polar molecules. You can separate them very well based on how strong they are. And always remember, anytime you use elution solvents and TLCs, that solvent levels never can come close to those of your spotting dots, never, because that'll ruin the whole thing. It'll pull your compound into the solvent itself, and the whole thing will be messed up, and all that work for nothing. So make sure that solvent level is well below your spotting line. So this lab, as you can probably tell by this point, is 99% setup. It's, you know, getting the plates, marking the plates, pencil lining the plates, setting up your solvent chamber, spotting the plates correctly, eluting the solvent slowly up the TLC plate, you know, tracking your dots, making sure the solvent doesn't go all the way to the top, taking it out, letting it dry, marking your pencil lines, checking with UV to see if you have any invisible spots or not visible in normal color range. So a whole bunch of that. So the analysis part at the very end, it's very quick. So that's the good part about it. So you got to let your plates air dry, because you don't want any liquid solvent, because that stuff might not be fully moved. So you want it to be fully air dry to stop any movement. The key here is not to use heat. Both the silicon dioxide and the alumina don't like heat. And you might have some reaction with your spots. It might remove the spot completely. So never use heat. Just let it air dry. Just basically put it on the watch glass with the white powdery side up. And then, of course, examine all your plates under UV lamp to see any of the elution things that might not be normally visible. And you can always circle them with the pencil to show where they are if you can't see them with your naked eye. And then you're going to calculate your RF values, and remember that's just a retention factor. So it's comparing the distance from the start point to a spot divided by the distance from the start point to the top of the solvent line, whatever that was, however high up your TLC plate went. And you would calculate and record each of those values for your unknown and your multiple knowns for those, and you can use those as a reference. So if you get, for instance, a 0.4 retention value for one of your compounds, if you have another compound that has a 0.4 and using the same solvent, it's going to be that compound. So you know right off the bat. And then based on all of the stuff you did and their retention values, you can look at your unknown and see however many dots. You'll generally have 2 to 3 dots. and they'll line up with some of the other dots, or if there's the dot on a different plate, because you can't compare plate to plate, that's where the retention values come in. If you have a dot that matches the retention value from a different plate, you know instantly that's going to be that known compound. So you would just use a combination of retention factors, and if it's on the same plate in the same exact location, then you can say you would know the comparison of your known to unknown. All right, so just the last iteration of some pretty essential concepts that you'll surely be asked about. Remember, don't touch the face of any of the plates with your fingers, glove or not. Always use a little clamping action with your forefinger and your thumb to grab it from the sides, always. Only use a pencil anytime you mark those plates, and make sure you mark it lightly and very gently, because if you use any pressure, you're going to dig into the silicon and you might have the aluminum foil or the glass underneath exposed, and that'll stop the solvent from going up. So you don't want that. Always light and always only a pencil. Make sure the solvent line doesn't touch the starting point or your spots. Make sure it's well below that. And also, as it elutes up, make sure it doesn't go all the way to the top, because then it'll start fully saturate and come back down and drag the lines down. We don't want that. So never have the solvent touch the dots at the base. And then when it starts to elute up, make sure it doesn't go all the way to the top. And then once you're ready to start doing it, once you put the plate inside of your solvent chamber, make sure you cover it with a watch glass while it's eluting. Make sure that it's an adequate sized beaker. You want to be able to get your hand in there and take it out quickly if you need to, because some of the solvents might go fast, they might go slow, but you never know. Now, safety is always a concern when you're doing TLC, because you're going to be using a lot of solvents and a mixture of solvents. You might get a lot of carcinogenic solvents, reactive solvents, lacrimators, or solvents that smell like rotten fish. And so there's a lot of things that can be bad. So anytime you're doing a TLC, the key thing to remember is you always have to work in a fume hood. Always. There's too much risk for problems. Always avoid any type of inhalation of fumes, because we could use some bad stuff in these solvents. So always watch that. Of course, as anything, always wear your safety gloves and goggles. And the one good thing to do with labs like this, when using a lot of mixture of compounds you might not have ever been exposed to, is always check out the MSDS, the material safety data sheets for your compounds, because they'll be given to you in labs, and they show the toxicity levels, any flammability hazards, shock hazards, stuff like that. So always check those out. And then again, if you're concerned and you're unsure, always ask your TA, your instructor. We'll be able to tell you.